Okay. And I'm also going to give you record access. Okay. I think that works. Okay. So, well, we are in week 9, which is pretty amazing. And again, thank you for all kind of sticking with us. So I just want, before we get started, are there any, I noticed that there aren't that many submissions for part 8. Are there any questions about part 8 before we get started? And you don't have to, you can just message me in private if you'd rather not, if you'd rather be anonymous. How are people feeling in general? Are you feeling stressed out, or like, is there anything I can help with? Yeah, it's the time of the year. Yeah, end of the term stress. Okay, well, it's good to know how everyone is feeling. So we'll try to take it easy today. I have to admit that, you know, I'm not immune to stress. I actually kind of had a mini meltdown last Friday, which is why we didn't have office hours. So I do sympathize with all of you. It's not been an easy year for anybody. So anyways, I'm trying to be like, as you know, accommodate for everyone. Okay, so it seems like there's a couple people who want an extension on the assignment. Happy to grant that. If you want an extension because you're feeling stressed, just please let us know. You can email Eric and just let him know that you need an extension, and we'll keep up the things. So okay. So let's, so a couple of announcements. So today we've got presentations from Brady, Brian, Colin, and Sarah. Thank you very much, Brady and Jessica and Lindsay. That's and Jessica. So that's really nice to hear. So those are the 4 presentations I have today. Am I missing anybody? This is everyone that I have listed on the sign-up sheet.
Hi, this is Casey.
Oh yeah.
Yeah, that's okay.
Sorry, Casey. So
no worries at all. Thanks.
And then let's see. So let's talk about kind of the final, the rest of class. So I want to talk. So for the final project, I thought what we would do is that we would look at a data set together. Who here has heard about Nhanes? Have you worked with the Nhanes data set at all? Ah, okay. Well, my thought is that we actually do some logistic regression on Nhanes. And I'm going to give you like the choice of 3 different outcomes. So like, I just want to kind of, let me share my RStudio and I can kind of show, I haven't finished the assignment yet, but this is going to be, this is in the part 9. Let's see. It is part 8. I can't read. No, that's not it. Okay, it's in the RStudio project. Sorry. So if you're in the Part 9 RStudio Cloud, so what I've done is I have, this is just kind of the pre-work. I will publish the actual project up, like I'll try to have it up by tomorrow. But the basic idea is that we're going to be looking at an extract from Nhanes. So this is called the Nhanes data package. And if you look at it, you'll see that there are a lot less variables than in the entire Nhanes data set. I believe there are something like 1,000 variables that were measured in Nhanes. And so you can see there are a number of them are categorical variables. So the ones we're going to concentrate on are diabetes, depression, and sleep trouble. So these are going to be 3 outcomes that we're going to model using the Nhanes data. And then, so part of this is like, I want to make this kind of an open assignment, and I want to make this kind of a collective assignment. So I want us all to kind of look at different combinations of variables in our models. And we're going to kind of compare like the predictive power. So number one, this is not a contest. It's not a contest to find who gets the best predictive model. It's really for kind of collective, our collective knowledge and to understand the data more. So that's the skim. So for this week, for your assignment, I want you to pick out one of these 3 outcomes to model. And then you're going to have to, if some of these outcomes have multiple values, for example, depression has none and several episodes of depression and most episodes of depression. So what you'll have to do is basically kind of binarize them by combining categories. So like for example, you could use case 1 to keep none as none and then depressed to collapse both several and most into a category. It may be easiest to make this into a Boolean variable, and you'll see that when we get to the logistic regression. So what I want you to do is also kind of look through the data and pick covariates that you're interested in as using predictors. One hint is you may want to assess correlations between variables. So obviously there's some variables that have similar names in here. So like, for example, all of the blood pressure ones, you may not want to include all of them. Or urine volume 1, urine volume 2. These were actually collected at 2 different times. So just kind of keep that in mind. So what I want you to do, and basically as part of this assignment, I want you to basically do your EDA and just kind of show your justifications for picking your predictors. So there is a Google Sheet right here. So I want you to, I'm just going to open it up. And so the way I'm thinking about this is, well, right now, just put your name and pick an outcome. And so the 3 outcomes are going to be kind of 3 different analyses that we're going to kind of combine together and kind of get some insight into this data set. And what I would like is each of the groups, each of the outcomes, to kind of talk amongst yourselves and decide which predictor variables each person wants to use. And what I'm going to have you do is build like 2 different models with 2 different sets of predictors in that set. Does that make sense? I'm not sure I completely explained it well.
Just so I understand correctly, is this for what we're doing in the class today, or is it like for the homework assignment?
So this is for the final project. So I just want you to.
Oh, okay, yeah.
Yeah. I just like,
I asked Eric this. So sorry. I asked Eric that is this separate than part 9 homework assignment, or is the final the same?
So I'm making this the part 9 homework assignment, but the final project. So I think I'm not going to have a part 10 homework assignment either. I'm just going to have the final project, because I just want to be fair to you all, because I know you're all stressed, and I think, you know, doing this project is you're going to be able to kind of apply these concepts. Does that make sense?
So so we're going to have one project, not a part 9, but a final for this week and next week.
Yeah. So for that's what's confusing. Part 9 is part of this project, but you'll submit it as a part 9 assignment. So this is in the part 9 folder. So just submit this as part 9, and then there'll be the final project. So there won't be a part 10 assignment. Does that make sense?
Do you expect us to collaborate for the part 9 project as well, or just the?
Well, it may be helpful to kind of talk, you know, like it's probably best to do like the EDA on your own and kind of bring it and talk in your group about how you're going to divvy up the covariates. So what I will say, yeah, so your goal is really to kind of decide on a strategy of covariates you want to use in different models, and each person is going to run at least 2 models. Does that make sense?
Yeah, thank you.
Okay. So this is yeah, go ahead.
Sorry, Todd. Yeah, I was just trying to make sure. So like for the final, then would it kind of be that everyone's just compiling their different models together, because everyone has to do 2 separate models? But
yeah, so in the
like
end, this is going to be like a score. It's not a scoreboard. We're going to report our predictions using these logistic regression models. I am going to make as an option, like if you want to apply a machine learning model, which we'll be talking about next time, you can do that for some extra credit, but I'm not going to require that, because I think the more I kind of go through the tidymodels section, you know, the more work I think it needs. So I'm not going to require machine learning as being a part of the assignment. It's just mostly to be running the logistic regression. So Sarah's question is, so basically we choose an outcome, and based on that, we will select predictors. Yes, exactly. So some of these predictors are not going to be probably useful for some of these outcomes. That makes sense. So part of the assignment is to kind of think that through. So does this sound like interesting for everyone? I do this in other classes. I do this in the analytics course, and I think it's always interesting because, and this is like a real life use case. My friend Eric Fezco actually sent me this paper in which they had something like 20 different groups look at the same neuroscience and publish their analyses. They were all different. And based on kind of your reasoning, like I'm getting an internet connection, you know, if I'm not coming through. So based on kind of your reasoning, like, you know, something like there's lots of different paths. And so I think exercise everyone, and hopefully we'll have some interesting results. So I try to do these kinds of assignments because I think it is interesting, because you have a part that you're working on your own, but you're contributing to a larger thing. So One thing I might do is I might do like, might have some sort of write-up of the final results. And I'll have to think about that. Maybe we can do that on, I'll leave it open whether you want to meet on finals week or not, but that's probably what we would do finals week, is just kind of go over results. I'm going to push doing a web page over to then, because I'll make that day optional for people who just are stressed out and they don't like, but so next week is going to be kind of the last class. So is there a data dictionary for the Nhanes data set? So there is a really limited one if you just do !Nhanes. So this is just a subset of Nhanes, but so it's incomplete, but at least it has most of the information you need. So just to get there, you just need !Nhanes. And so, and I fully admit that this is probably not, I'm probably going to have a bunch of survey people yell at me that this is not the point of Nhanes, but I think it is interesting at least to kind of ask these questions. To me, it's a fascinating data set. I've looked at it for probably 4 or 5 years, and I still find interesting things about it. So, okay, any other questions about the final project? So I'm just going to go over. I'm going to go over muddiest points. And why don't we have 2 presentations, and then we'll take a break, and then we'll kind of finish up. Yeah, we'll finish up with the next 3 presentations after that. Okay, in terms of muddiest points, so yes, these are all valid. I still find PER very confusing. So, and a lot of the comments were about nesting and how it still kind of is a little mind bending. So I kind of ran out of time. I still want to show you that crop yields analysis that I did. And I will show that uses nest and unnest. So this was a point. So per data frame model tables. So the point was made that it is what I showed you was a way to apply one model to a bunch of data frames. I'm going to end the class showing you how to specify multiple models and apply it to a data frame using PER, and making using list columns. So are there any questions about this before we kind of move on from this? Did the homework help, or do you still have questions? Okay. So just want to thank, these were nice comments. While the nesting and nesting were more challenging topics, I thought the examples were great. I felt pretty good about everything by the end. Lots of love for GG pairs. I like it too, and it would have been useful for the biostats project. Yeah. Again, I'm sorry that wasn't more timely, but I will pass this feedback on that that at least Tuan should show you GG pairs, because it is such a useful visualization. And thanks for teaching cool stuff. You're welcome. So this is a point that is well taken. My brain is always so full by the end of class that I forget what was fuzzy and what was clear. Point taken. Oh, well, I'm glad that PER helped you with ANOVA, the ANOVA assignment in 512. Okay. So why don't we have 2 talks now? So actually, I think we should have time for 3 talks. Who wants to go first?
I can go first, if that's okay.
Okay, Casey, let me. Okay, so you should be able to share screen, Casey.
Okay. Thanks. Sorry if there's any like wind background noise. I'm outside right now.
Yeah, no worries. We can hear you.
Okay. 1 second. All right. I think everyone can see that. So
Yep.
I will be doing an overview of Str Detect, which is a function within tidyverse, specifically the string R package. And so what it allows us to do is look for lines meeting, or look for strings that meet a certain pattern within our data. So we can look for strings that start with a particular character, contain a specified number of characters, like number of number of multiples of characters, or a set of a few different ones. We can also combine it with other functions to filter data by the results we specify, or to look at totals or proportions, which can be useful when looking at larger character vectors. And honestly, you can do a lot, which when I was initially looking at the function, I got pretty bogged down. So I met with Ted and I was like, yeah, so I outlined like 5 different examples, but it was a little much. So I linked this PDF, which kind of visualizes some of the other functions within the string R. And so in this first example, I looked for islands within the penguins data that contain the letters T or B, not in like a specific place, which is denoted by the vertical bar, so T or B. So the output shows us basically it went through every row within the islands and said true or false whether or not it had T or B in it. So basically we got the Torgerson and Biscoe Islands, but that's kind of, cumbersome to look at, doesn't really help us a lot. So I used the same idea, but with the caret, I denoted that I was looking for strings that started with T or B and then made a subset of the data. And so now that'll contain all the Torgerson and Biscoe ones. And so overall, I would say it can be really helpful when working with text data or strings. I guess not as much for the penguins one, but I found a really helpful example online where it was like a blog post and the author had 6 different cities in his data set about airlines that contained the phrase news, like different cities, but he only wanted to select New York and Newark. So using, this code down here, so it's new and then the dot and the asterisk, kind of do something where basically he was able to filter it. So it's like, I don't know if I didn't really specify what the dot and asterisk do for this one, but basically he was able to filter it and then get counts for those cities in that way. So overall, I think it can be really helpful for certain kinds of data sets. That's all.
Yeah, I will say that Str Detect is probably one of the deepest functions in the tidyverse. So like great job kind of summarizing that, Casey. Does anyone have any questions for Casey? And so the specifications, the way you kind of search is it's called a regular expression. And this is kind of way beyond this course, but so that dot and star are kind of like wildcard characters. So the dot, I believe the dot matches any character, and then putting a star after it means, and I think it's any number of times. Do you remember, Eric? I can't remember just offhand.
I think that was it from what I remembered reading. So
yeah,
I just didn't write it.
yeah. So the notation, this is something that it's powerful, but there's some learning that kind of is attached, but just for searching different kind of phrasings, it can be really helpful, and you can use that in your filter statements. Okay, who wants to go next?
I can go next. This is Brady.
Okay.
So let me share my screen. Okay, can you see this?
Mm-hmm.
Okay, so I did geom density from ggplot2 package. basically what it does is it makes smoothed histograms of the density of numerical data. I think it's mostly used for continuous data, but you can also use it for discrete. It's just not as good. But what it's really useful for is for looking at the distribution of your data, and you can use it to look at the skew and kurtosis as well. And what I really love about it is that in ggplot, it just has like, when you use it with a categorical variable, it has really good colors already. And so this was, but I just did a ggplot with this density with penguins data. I did the body mass by species of penguin, and you can kind of compare just the different distributions. Like Gen 2 looks like it has the biggest body mass, and chin strap looks like it's pretty equal with Adelie. And then inside of the function, you can specify your alpha, and I think it goes from 0 to 1, and that's the opacity of the color. And so that's super helpful, because if you have multiple different density plots in the same graph, it can get really confusing if they're not clear, like you can't see through to the other ones. So I thought it was really helpful, because especially when you're doing exploratory data analysis, because it's really helpful to not only do descriptive stats, but also see visually what you're looking at. Yeah, and that's all I have.
Great. Thank you, Brady.
Uh-huh.
Any questions for Brady? Oh, and Eric posted a link to this website called regex 101. This is, I use this too. But anyways, any questions for Brady? Yeah, I would say geom density is one of those functions that's just, it's really nice. If you, there is kind of the, I can't remember what the 2D version is, but I use one called geom hex, and that's for 2D density plots. So those are, that's also a really helpful one. Okay, how about who wants to go next?
I can go.
Okay, go for it, Colin.
Okay. Are you seeing both of these RStudio windows?
Yep.
Okay, I'm gonna do it in this rather than in the knitted document, because I'm gonna be making files. It's like impossible to see what this function is doing in the knitted thing. So I had per walk, which is in the map set of functions. So we're taking a list and applying a function to everything in that list. It's just different in the way it handles what gets returned by the function and like what happens to the side effects. So I have a couple examples where I'm comparing it directly to map, so you can see the differences. So first, I had the Palmer penguins data set. I split it up into the species, and this function just makes a plot. So when you run map on that, you get a list. Whoops, let me get this library up. So when you run that, you get a list of 3 plots, just like we would expect. And the object you get back from map is a list, and it is 3 things long. So from walk, if I tried to do that same thing, nothing would happen, because that's just not really what this function is for. So if I try to do this again, but take this object back and like actually have the output, I get back the list that I put in rather than the plots. And that's kind of the core of what this is supposed to be for, I think, is that when you put it in like a big piped structure, it's kind of like a save point where you can like write to your disk or something, and then keep what you put into it and keep going with it. So here I'm just writing these individual data frames as CSVs. So when I run this, you'll see they appear over here. So now I have a CSV for each one of the species. We're going to delete those. But it returns a list of the same length, but all the indices have nothing in them, which is kind of weird. So when it's not pointing to anything, you get an output that is kind of useless. Or I mean, maybe you can use a list that has 3 empty indices for something, but I just don't know what it is. Then if you do make this map function, or this is map. Right. So if this points to something, what you get back is not the same data type you put in, but basically the same. And then if you do walk, it would, let me delete these so you can actually see that happen. When you use walk, you don't get any like annoying output that like 3 nulls. It just saves the thing. And then if you point to something, you get the list back. So is that helpful? I struggled to think about why one would want this until I remembered this figure that I put up in lab meeting recently for a program I built in Python that talks to Fiji a lot, which is like an image processing thing that some of you might be familiar with. So for something that you needed to talk to other programs and save a lot of files in a systematic manner, this would be really helpful. This would have saved me a whole lot of time last year if I knew what this was then. So yeah.
Great. Thanks, Colin. So yeah, walk is one of those functions, as Colin says, it doesn't seem that useful, but oftentimes when you're using it as an entire workflow, so if you wanted to save a bunch of plots, for example, like it's really useful for that. So you could basically do a per walk and then like a GG save and basically save all of the GG plots in a list. It's also, yeah, and for writing files. So if you had like intermediate kind of results, like as Colin said, it's really helpful for doing that. And it's in the middle of the pipeline. Oh, and thanks for posting that link, Brady. That looks really cool. Okay, let's see. Why don't we just power through with the last 2 presentations and then we can take a break. Who's next?
This is Brian. I can go.
Yeah, why don't you go for it, Brian?
Okay. All right, so for my short presentation, I decided to do pluck, which is in the per package. So what is it for? So I think we've all seen throughout the last couple of weeks how complicated the syntax for lists can become. And so pluck just gives a sort of an alternative, more intuitive way to retrieve specific elements from lists. And so I just created a simple list. And within that list, I have 3 lists, which contain movies, release dates, and then just personal preferences. And so we learned before that we can use this bracket syntax to retrieve specific elements from within the list. And so this says we're retrieving the 5th element within the first list. And so we get this movie title. So this can become really complicated really quickly, especially when you have nested lists. And so pluck sort of gives a more logical and easy to read approach for retrieving list elements. So for example, for the same code, we would do pluck example list 1 element 5, and then we would get the same result. And then you could just keep going from there for each sort of index. You could just keep going and going for the nested elements. The really nice thing about pluck is that it consistently returns null when an element does not exist, which is not the case for this bracket syntax. And I think we learned about why null is really nice in one of the lectures. I think it was called like coding to fail. And so that's a really nice element about pluck. There was a lot about pluck and then chuck, which is essentially the same function as pluck. It just, its default is that it will report an error when an element does not exist, and then pluck will not. So you kind of have those two options. Another thing that you can do with pluck is that you can sort of apply it to the map function that we learned about, and then retrieve specific elements applied to all of your lists, which is really powerful. You can also assign specific elements, or assign an element to a specific spot within your list. So like I assign this release date to spot 2 in list 2. And then you can also assign different functions within your list. So you can do like a user-made function and apply it to pluck. You can do any of the common R functions like max and mean. And then just the very last point that I wanted to make was that pluck is not just restricted to lists. So you can use it on like data frames. And so I had this data frame about like shark attacks. And so, for example, if you wanted to know what species of shark attacked a specific case number, you could actually use pluck to answer that question. So you can make an index for the specific case number and then apply that index into the pluck function. And then having your species, that's the column that we're looking at. And then when you apply that function, you see that a tiger shark attacked that case number. So I think that pluck is very helpful, and because it's a more intuitive and more powerful way to actually sort of get specific elements out of a list, and it's easier to read than the bracket syntax. And then you can apply it to different powerful functions within per, like the map function. So yeah.
Great, thanks so much, Brian. Yeah, so pluck is one of those functions. It's surprisingly powerful, but as Brian said, anything that prevents me from having to write the double bracket notation, I like, because I always forget a bracket somewhere and my code breaks. So thanks. Okay, who's the last person? And then we'll break for a 5-minute break. So Sarah, were you presenting today, or did you present last time? I can't remember.
Which Sarah?
Sorry, Sarah S. I think you presented last time, right? Sarah.
I think she presented last time.
Yeah. Okay. So we're actually good. So why don't we take a 5-minute break? And we'll come back at 4 o'clock. So if there are any questions, just let me know. I will pause recording. Thanks. Okay. Okay, so we're going to be talking about a little bit about tidymodels today, but we're also going to be talking about kind of unsupervised learning. Has anyone used principal components analysis, PCA? Okay, I'm getting a no. Okay. Okay, so this is good to know. I mean, so this is a really useful visualization technique, and we'll get into it in a second. And it basically kind of allows you to kind of condense lots of different variables and of data into kind of two axes. And I'll talk a little bit more about how it does that. But it's a very useful visualization tool and data exploration tool. Okay, let's talk about today's learning objectives. So I just want to talk a little bit about the different types of machine learning and kind of some distinctions between machine learning and statistical modeling. So we'll talk about how to split data using the R sample package. We'll be learning about some of the standard approaches for pre-processing data using this package called recipes. And in machine learning, there are kind of these standard steps that we kind of apply to kind of any data set before we kind of run a machine learning algorithm on it. We'll learn about PCA as a visualization and exploration technique. Sorry. And the last two objectives that's actually going to be for next time. So what is machine learning? So one definition I found was the study of computer algorithms that improve automatically through experience. I don't find this to be a really useful definition, but I like the second one, using algorithms to find predictive patterns in data. So there are kind of three, this is kind of, to be honest, I don't even know if I would put unsupervised in machine learning, but it is called a machine learning method, so I'll talk about it. So there are kind of three different types of machine learning algorithms. The first is called unsupervised. And this is about discovering groups in data without kind of knowing anything about them, except so if we don't if we have a bunch of measurements and we're just interested in seeing how the data is grouped, we can use these unsupervised methods. So like I said, dimension reduction, such as principal components analysis and clustering, are considered kind of unsupervised methods because we don't really provide it any information other than the data. The point about unsupervised is that the goal of unsupervised learning is really discovery and exploration, not necessarily prediction. So if I find, two kind of natural groupings in the data, it's up to us to basically kind of follow up and understand why the data is kind of grouping that way. So that's in contrast to what's known as supervised learning. So we're actually providing something called a label to our algorithm. So, in some cases, you may have seen there are some really kind of unethical applications of this where basically given someone's prison record, can you predict whether they're going to be recidivist or not? So that kind of basically we have a data set and we kind of know the truth. And we're trying to be able to, given new data where we don't have a label, we're trying to predict the label for that person. So in that case, we have a data set of recidivist versus non-recidivist prisoners. And then we basically cycle through both of these cases, trying to learn information that helps us kind of predict whether someone is going to be recidivist or not. I'm not very comfortable kind of talking about this example, so I'm going to shift because that's just kind of an unhelpful, I think it's an unethical kind of application of this. And we'll talk about why in a second. So the idea with supervised learning is you're trying to predict whether something is a particular particular, like, has a label of something using all of the covariates that are in your data set. There is a kind of third type of supervised learning. I'm not going to talk about it. It's called reinforcement learning. And it is kind of it kind of builds upon supervised learning, in which you're basically kind of guiding the algorithm and telling it kind of what kind of solutions you're interested in. But like I said, today we're mostly going to be talking about unsupervised. I just want to kind of introduce these kind of three different types of machine learning. So next time we'll be talking about supervised learning. So this is really contentious. I've been trying to really find, like, you talk to data scientists and they will say machine learning is very different than statistical modeling. But I just want to kind of contrast it. And in this case, I'm talking about supervised machine learning. So statistical modeling, the goal is really to understand and quantify relationships between covariates and outcome. So, like, for example, in linear modeling, we want to quantify the linear relationship between a covariate and our outcome. So oftentimes in like linear regression, you'll say that for every unit increase in my covariate, you'll see this much increase in my outcome, or decrease, depending on the relationship. You can predict with a statistical model, but really the prediction is secondary. The primary goal for statistical modeling really is to quantify these relationships. The other thing to keep in mind in statistical models, our sample sizes tend to be smaller because a lot of the statistical machinery is made for actually really small sample size cases. This is kind of in contrast to machine learning. So this is kind of a subtle thing, because you can use regression in a machine learning context. But the point of the machine learning application is that your prediction is a primary goal, and understanding the relationships between the covariates and outcome is kind of secondary. The other side of it is that the sample sizes when we use machine learning tend to be much larger, usually on the thousands to hundreds of thousands to millions to billions. So that's kind of the other side of it. In terms of logistic regression, so I will say you can use logistic regression either as a statistical modeling technique, and I am sure someone in SPH is going to get mad at me for saying this, but you can also use it as a machine learning technique. But it's really, so it's the same underlying modeling, but your purpose is different in both cases. Any questions about this? And we'll talk more about this next time, but I just really want to kind of start drawing the line between these. So let's talk about tidymodels. So tidymodels is basically a set of packages like the tidyverse, but it's really focusing on machine learning. I will say that there's a lot of functions in here that are very helpful for statistical modeling as well. So there are these very kind of high octane machine learning packages. Has anyone heard about Tensorflow? Yeah, so Tensorflow is a machine learning package that lets you produce what are called neural nets on a data, use a neural net algorithm on a data set. It's one of these packages that's kind of used a lot. We don't cover it here. If you're interested in this, there's actually a deep learning, class in DMICE, my department. There is a machine learning class. They do suggest that you take that before you take the deep learning course. But the point of tidymodels is to learn one workflow, and you can swap out many different machine learning algorithms once you kind of learn this basic workflow for tidymodels. So this should be the different packages of tidymodels. So the different packages of tidymodels are designed to be useful in a tidy workflow, and they roughly map to the different steps and requirements of a machine learning workflow. So I think the next slide. So these are kind of the basic packages. This is just really for your reference, but I'll be talking about this in terms of the overall flow. So there is the RSample package. And this is like if you need to do something like bootstrap sampling, or you need to split your data up into what's called a test and training set. And we'll talk about that in a second. RSample has a lot of functions to do that. So it's an extremely powerful package, and depending on how you need to kind of structure your samples for your data, your resamples for your data set, it can be an extremely powerful package. The recipes package, it took me a while to really understand what was going on in recipes, and we're going to be focusing on recipes today. So these are the functions to pre-process your variables, and if you need to numerically scale them or normalize them, there's lots of functions in there. And so there are a lot of functions in here for kind of transforming your data. This is also, this kind of pre-processing is also known as feature engineering. Max Kuhn, who's kind of the primary architect of tidymodels, has a great website, and I'll post basically that talks about feature engineering. Parsnip. So this is what basically this lets you kind of specify your model structure and training your model. So workflows. So a workflow basically lets you integrate both a recipe and a parsnip model into a single workflow and use them reproducibly. We'll get into that next time. The final package that's really useful is called Yardstick. So you use these functions to evaluate things like the accuracy of your predictions on data sets. So this is kind of the general machine learning workflow, and we'll go over through each of these steps. So we're really mostly going to be focusing on these first three steps here. So the first is the point is to get your data, and then you do the pre-processing and transforming of your data. And these transforms can be like transforms that you are familiar with, such as box-Cox transforms or log transforms, for example. Once you have pre-processed the data, you need to split it into, and this is machine learning, this is kind of machine learning 101, is that you train your model on a set of data called the training set. And we'll get into that in a second. And then you test your model, you test the actual predictive power of your model on a test set. The main point really is that you're not double dipping. So like, if you're using this training set here to basically fit your model, if you really want to evaluate the predictive power of your model, it has to look at data it's never seen before. And here, then we basically start actually doing the model fitting. So that's kind of this section right here. We need to specify what kind of algorithm we want to use for a model. We will basically we'll select what are the informative features or covariates in our model. We can do this either automatically or by talking to our collaborators and getting an idea of which variable covariates in our model are more important. Once we have that information, we can train our model, and then we can evaluate the predictive power of our model. And so again, we train our model with the training set, and we test the model with the test set. That's a lot. And again, this is just to kind of introduce you to the basic workflow. Like, we're going to focus on the first 3 steps. But the point of this showing you this workflow is just to show you what parts of the workflow kind of correspond to which packages. So you can see this is like recipes, this is R sample, this is parsnip, and this is Yardstick. Any questions about this so far? Okay, so I am going to talk and one of the things about kind of machine learning and tidymodels is it seems like they basically basically replicated dplyr, but it's kind of different. So I want to kind of start you off with, this is kind of the basic structure that tidymodels kind of expects of your data. So basically, it's like a data frame, and just like we've encountered before, our columns are features or covariates. The main difference is that we've got an outcome. This can be a continuous variable, but in our case, we are going to be working with dichotomous variables. Basically, it's going to be, so like in that previous example, it would be recidivist versus non-recidivist. The prisoners, this could be, in our case, it's going to be, is a penguin Adelie or not? So you can begin to see that we need to start kind of transforming our data. So just to kind of go in further, so our labels are the thing we want to predict. So I made this slide for the neuroscience class, so I'm sorry these examples aren't very kind of stats friendly. Anyone have something that they would like to predict that's a dichotomous variable? I mean, this could be like, this could represent, like, this row could represent cats and dog, and like, so this label could be cat versus dog, for example. So we have all of this information on our animals, and we're just trying to use that information to predict whether it's going to be a cat or a dog. Okay, I think I'm going fast. How is everyone feeling? Okay. Okay. So this is talking about splitting the data into training and test sets. So what we do is we randomly sample each of these rows and we split them off into our training and test set. So you can see that, like, everything that was marked as blue kind of goes into our training set, and everything that's marked as pink here goes into our test set. We know, and the thing to note is that we know these labels, but we want to see what the algorithm thinks. So we're going to compare these to the algorithm's predictions to our actually the real truth. The other important thing to think about is that the proportions of labels in our test set and training set have to be roughly the same. And this is important. So like, for example, this has kind of 5 and 5 here. But like our training set is a little bigger. And so you can see we've split our labels up equally. So if you, and this is what can be difficult when you have something like a case control study where you have so many more controls and cases, there are approaches that you have to do to kind of deal with that. So there are kind of resampling based approaches that we can use to deal with kind of case controls. But one of the issues is that like case control studies, much fewer numbers in the cases compared to controls, but we make we have kind of ways to kind of deal with that. So this is the this is the function called initial split, and we'll be talking about this in a second. So you run initial split on your data set, and so this is called all features. And the proportion is the size. So is the size of the training set. So 75% of our data is going to go in our training set, and 25% in this case is going to go in our test set. So this is really this. And these numbers, so people always ask me, so is this like a fixed number here? So it depends on how big your data is. Oftentimes, you know, our test set has to be very small because we don't have that many rows of data. That's kind of a nightmare. But the usual rule of thumb is usually about 8. If you have a lot of data, about 80% of your data should be used for training your algorithm, and then 20% of your data should be used to test the algorithm. Okay, so and we'll encounter this when we actually get to the lab, but I just want to take everyone through the process so we'll be familiar with this. So like I said, recipes basically outlines approaches to transform data using using these functions called that are just their step and then underscore. This is just the link to the tidymodels page. So here a recipe starts out with specifying the recipe function. And then here, like again, we're going to use formula notation to specify our outcome. This is something that you might not have seen before, is this dot notation here. This dot notation means everything else other than species. So species is going to be our outcome variable, and we're going to include all of the other covariates as predictors. You need to pipe that into these different functions. So there is one called update role. We'll get into roles in a second, but the point is that you can apply these different kinds of transformations based on the data type. So if it is numeric data, then we might want to normalize it, which is just kind of apply scaling to the data. And there's a lot of reasons that like we need to do this. Some algorithms need to have our data normalized to a specific range for it to work. So let's just talk about data types. So this is just my kind of extension of the penguins data set. So we've got a penguin name, so Gary, Flo, and Bernice. Species, and built, like, you know, we've got numeric data, and then we've got categorical data. This is what kind of drives me a little crazy about tidymodels, is because the wording for these is just similar enough to regular R data types to be confusing. So let's talk about what these data types actually are called in tidymodels. So the ID type, and sorry, my handwriting is terrible. The ID type is basically, it's not used at all. So this is like if you've got particular identifier columns in your data set, this basically, calling it a type ID basically says don't process these columns. They're just for identification. Our blue one is our outcome. And so, and then the more important ones are numeric and nominal. So numeric is basically any, and this can be integer, this can be continuous values. Nominal is what tidymodels calls character. So like I said, Recipe has many standardized steps for processing data in machine learning. Here, this is some of them. So you can see like we can do things like impute missing data. So this is just kind of, I just tried to kind of summarize what data type this and what kind of functions you can apply to do these kinds of processing. So imputing missing data, there's another function that will let you do imputing, but this one is really helpful. It's called KNN impute. And so you're looking at the values of its neighbors. And we'll talk about what that means in a second. But then you basically say, okay, you look at like, you know, like 3 of its neighbors and look at the identity of that, and you basically specify a value close to that. I didn't explain that very well, but there are other kind of ways to process data. So you can transform variables for skewness. So you're probably familiar with log transformation and box transformation. So you can do this transformation on data type all numeric. You can also put in the variable names, and this works just like the select function. So if you just wanted to process one variable, you could put in like the variable name, and we'll look at that in a second. This is very helpful, discretizing continuous variables. So there are functions for that. Like it talked about normalizing data, so centering and scaling. But it's also really helpful for categorical data. So you can automatically generate dummy variables. This one is, I haven't used it before, but it seems really helpful. So this is identifying highly correlated covariates. And basically, you know, like basically, so you can make decisions about whether to kind of include them or exclude them. If you want to create variable interactions, you can do that with a step interact. And then we'll finally close up with like this kind of doing the principal components analysis. Any questions so far? So you can see recipes is like an extremely powerful package for transforming data. And like I said, Max Kuhn is a very smart guy, and he's really kind of looked at what are all of the transformations that people do on data. So even if you never use the rest of tidymodels, recipes can be a very helpful package for you. So what usually happens is that we build a recipe once and we use it on different data sets. So depending on how the order in which you're doing it, you may apply a recipe to the whole data set, or you may split the data into training and test first and then apply the recipe to each of those sets. So there's not really any good guidance on this as long as you're separating the data out. So this is where we'll start moving into the lab. So just talking about principal components analysis. So thing I want to emphasize is that any of these kinds of summarization methods are really made for visualization. I see a lot of people getting into trouble by basically trying to make these into like something more sophisticated, trying to get more information about it. In general, what I'm showing you today are unsupervised methods. So we'll talk about this in a second, but principal components essentially, and I'm sorry for the misspelling, squish multiple, like if you've got like say 1,000 covariates, you can basically use this principal components analysis to kind of squash them into two kind of uber variables that kind of represent all of those variables. And we'll talk about that in a second. And so the way it does this is basically it finds linear combinations of all of these different other variables and to produce this uber variable. And there is kind of this approach, the values of these like combinations are chosen to maximize the amount of variability that's covered in each of these axes. So usually in the case is like, we've got, and like, for example, in flow cytometry, like, we have 90 different proteins. Someone wants to try to make some sense. Is there a kind of a separation between groups? And we'll look at this in a second with the penguins data. But so the principal components analysis lets them kind of summarize the data visually and see, and basically kind of in a way to quantify how difficult it is to separate out our two different groups, or in our case, our outcome. So this is just, we'll go over this in a second. So the other unsupervised method I'm going to show you is clustering. We'll go over this when we get to it in the lab. Finally, we're going to end up with logistic regression. So any questions about this before we kind of leap into the project? And like I said, this is usually a full course. So, just like if this is confusing, just kind of go over it and definitely come back with questions. So let's see. Let's talk about like kind of learning objectives for today. So we're going to utilize the resample package to produce our test and train data sets. We're going to understand how the recipes package makes pre-processing reproducible. We'll apply a recipe to a data set to pre-process it. And we're going to utilize data reduction methods. So we'll focus on PCA. If people are interested in this other one called UMAP, we can cover it at the end. And then we'll utilize k-means clustering to understand our data. And then we will apply logistic regression. So this is a nice, I've just been trying to kind of corral all of the different terms. So these are both, some of these are specific to tidymodels. Some of these are just kind of general machine learning things. So if you get, if there is a word that is confusing, I tried to basically summarize them here in this glossary. So just a caveat, so this is really only a basic introduction to a machine learning workflow. We actually cover machine learning in two of our courses, which is in the Statistical Methods course and in Shubo Song's machine learning course. So this is kind of a very deep topic. So, and like I said, there's a deep learning course here at OHSU. I talked about Shubo's course. So Melanie Mitchell is no longer at PSU, but there is a machine learning course at PSU that is less mathematical, but and very accessible. So, if this is interesting to you, that's another possible kind of route. Okay, so the first thing we're going to do is we're going to load up our old friend Palmer penguins. But what I'm going to do is I'm going to and actually, so we're going to select the numeric columns and the species for our data set. So most everything we're going to look today are just on numeric variables. So just keep that in mind. So what I'm doing is I'm just using the contains functions. So again, you can search on column names. So I'm just looking for the ones that contain millimeter and gram. And then I'm adding species to that. Then I'm going to subset species to just Gen2 and Adelie. And then we'll do this complete cases. So this basically makes sure that we have a value in each of our columns for each of the things. And then finally, we're going to, because species is a factor, we're going to drop the chin strap penguin. So there's only 2 categories in our factor. So I'm just going to apply this. So now just if you kind of do a skim, you can see we just have species. And again, species is going to be the outcome we're trying to predict. And you can see we have 151 Adelie and 123 Gen2. And you can see that there are, that we've got our numeric variables. So anyone notice anything interesting from the histograms, just kind of glancing at them? Yeah. Yeah, good point. Good point. So yeah, flipper length seems to be bimodal. So that's actually interesting. So that might tell us basically that flipper length might be a good predictor of species. So that's always why you skim the data. So if you get these hints of like these kind of bimodal distributions, it is usually a sign that your problem is actually pretty easy to do. But we don't know what the identity of these different bimodal bumps are. So we don't. That's just a guess at this initials. So here we're going to be using. So we'll do what like we said. So we'll do an initial split. So and that's on our selected numeric matrix, our penguins matrix. And again, we're going to use a proportion of 0.7 here. So that's going to be the size of our test set, is going to be 70% of the data. You can add here a strata. So this is something that you want to ensure kind of a variable you want to ensure even equivalence proportions across both the test and trained set. So in that case, we want species. Our data set is kind of roughly balanced. Our proportions are roughly similar, but we do want to, we can see that there are slightly more Adelie penguins than Gen2 penguins. We want to preserve that proportion across both of our test and training sets. So the first thing we're going to do the initial split. We'll look at penguin. And then from the initial split, we can basically get our training and test set using the training function and the testing function. So we know that our data set that goes into it has about 274 rows. So let's run this and look at the dimensions of penguins train. So you can see that penguins train is that's roughly 70% of 274. One thing I want you to see is, so if we do penguins train, and we do janitor table on species. So that's for the training set. So if we look at the testing set, so we've got about 54 to 45. You'll see in our test set, we preserve that proportion. Any questions about this? And you can, I think you can put multiple strata, but in this case, it's usually your strata argument is usually going to be your outcome. Okay, so now we'll get into recipes. So now we're going to process the data for use by our model. I think I mentioned this before. So recipes they always begin with a recipe function and a specification of your model. So this column, because it's on the left side of the tilde, is always going to be considered our outcome. And we're saying all of the other variables in our outcome are going to be predictors. And we need to specify the data set that it's coming from. So I mentioned this before when we talked about kind of the different kinds of column types and recipes. So outcomes, features, and identifiers. And like I said, we identified the outcomes and the features using the formula. So if you have like, in our example, if you had a penguin name column, you can basically make it into an ID column using this update_role. And just that basically makes, so it basically says, don't use that in our, as a predictor. It's just an identifier and it's not an outcome. So any questions so far? So this is our basic recipe, is just really here. And then we've specified, well, this does not exist in our actual data set, but this was kind of in our example. So we're just going to apply a couple of processing steps to our data. So step normalize. So that, again, that is about scaling. And these are other kind of transformations you can do on the data. So step dummy and step arrange. I've actually never used this. I think there are some algorithms that require the data to be sorted. I think that's what that's for, but I don't know just offhand. So like I said, because it kind of works in a tidy workflow, if you just wanted to step normalize like the bill length millimeter and bill depth millimeter columns, you can just pass those names into step normalize and it will only normalize those. And again, the difference between recipes and dplyr. So most everything that you're doing in recipes can be done in dplyr. The main reason why recipes exist, though, is that most of these operations are really for machine learning. Not all of them, but you'll see that most of them are. So I mentioned this before. So there are some handy selectors that you can use within these steps. So all predictors is a really useful one. So that's everything that is not a ID column or a outcome. So for example, we could run, like we start our recipe and then we can just use step normalize on all of the predictors. But these are the other, like I said, these are the other two that are very helpful. So all numeric, that's all numeric variables, and all nominal. So let's actually kind of do our recipe. Let's run our recipe. So this is really kind of annoying. So when you basically make a recipe and you pipe it through and you save it to an object, the transformation has not actually been done on the data. It's only when you use this prep function that the data is, the transformations are applied. And the data is a little bit hidden to you because you'll probably be like, well, okay, it's telling me that there's one outcome in my data set and there's 4 predictors in the data set. So the data is now transformed. But if you want to get the data out, and this is like the terminology that I really am not a big fan of, and I guess this was part of the juice or part of the whole idea of recipes having verbs like juice. If you want to get the actual transform data out, you have to use this juice function. And so you can see now our numeric columns have been scaled as we wanted it. And just to kind of make sure that that's the case, if we skim our variables, you can see that they all roughly have, well, they're scaled between one, I can't remember exactly how the scaling is applied, but you can see that this one ranges from minus 1.96 to 2.46. And etc. So this one starts at 1 and then it ends at minus 1.74 and it ends at 2.36. I believe that this is median subtracted and then it's scaled, but I'll have to double check. Yeah. Thanks for answering that question, Eric. So any other questions about juice? I really don't like this terminology, but unfortunately we're stuck with it because because Max decided.
Ted, I was curious about this juice function.
Yeah.
So I looked it up. It looks like it's been superseded with bake now, or like at least the bake function can do a very similar thing to juice.
Yeah, so there was the convention that I believe juice was for the training set and bake was for the test set. And last I checked, Max basically just decided to use juice for both the testing and training sets. And I think you can provide an argument called new data now.
Yeah, I think that's for the bake. Sorry, I don't mean to distract too much, but yeah, there's a way to use, I think, bake to do the same thing as juice here.
So bake is not going away, but just to make things simpler, I think Max wants juice to be applied to both. You can use juice on both the testing set and training set, from what I remember. I might be wrong, so.
Yeah, thanks.
Yeah. So again, we can apply our dummy. We can make a dummy variable of our data, of our outcome. So like if we specify step dummy and we use the all nominal selector, you can see now we have transformed it into one dummy variable. And so it is 0 if the species is not Gen 2 and it's 1 if the species is Gen 2. Okay, so we talked about building a recipe. So we're going to use it to do what's called a principal components analysis now. So again, we can apply our recipe to it. and we can apply step PCA to transform the all of our numeric variables with principal components analysis. And then we need to prep our data to actually transform it. So if you look at this, it doesn't seem like we did anything. We've got one outcome and we still have 4 predictors. But remember these like our new predictors are a set of variables that basically squish all of our data into kind of these a smaller number of variables. But in this case, it's a little confusing because we're putting in 4 variables and we're getting 4 principal components out. But we'll look at this in a second. So again, we need to, if we want to actually look at the data, we have to use juice on our prepped data. And so you can see that our columns are no longer called, you know, our covariates. They're now called PC1, PC2, PC3, and PC4. These are what are known as the principal components. In practice, you tend to only look at the first 2 principal components, but the analysis gives you these for free. So that's why you're seeing them. But for the most part, we're just going to usually look at the first, at the most, we'll look at the first 3 principal components. So, so this is just me plotting the first principal component versus the second principal component, again, with this data set. And then we're going to color it by species. And so this is the value of our first principal component. And again, we're squishing multiple variables into these these uber variables. And in our case, you can see that there is a pretty clear separation between Adelie and Gen 2 penguins. So this gives us a sense that there is basically a way we can kind of separate our 2 species in a way that we can predict them pretty easily. I want to show you a link to, I'm going to paste this in the chat. So I'm going to the bottom, sorry. This is Allison's, Allison Horst's thing about PCA and penguins. Let's see. So I'm just going to open this into a different window. So if you want to learn more about principal components analysis, this is kind of a nice discussion of it. So one of the things that you can kind of visualize of the principal components is the contribution of each of the variables to each principal component. So we'll get to that in a second. She had okay, so this is kind of what I was kind of getting at. So, and I've actually find these diagrams to be a little kind of hard to understand. But again, it's basically what's going on is that you're kind of squishing, all, like 4-dimensional data into 2-dimensional data with a very kind of standardized way of doing it. But you can see that these kinds of arrows kind of represent kind of the variable contributions to I think that, but let's get into the other visualization. But I just wanted to show that, like, you know, it's taking 4 covariates and it's squishing it down to 2. So if you just keep that in mind, that's kind of that's really what you need to know when you're kind of working with principal components. So I'm going to go back up here. Oh, and okay, so why don't we take a 5-minute break? It's 5 o'clock, so let's take a 5-minute break and come back at 5:05. Sorry, I wasn't looking at the clock. So there was a lot of confusion what these principal components represent, and I'm going to kind of talk about that in a second. But so this one is the first two principal components. I am just plotting the first and the third principal components here. So how do we actually select these linear combinations of principal components? So this is, I just want to show you what's actually done, what we're actually doing to kind of construct these. So this is, this basically gives you the coefficients for making the linear combinations. So the way to read this is that it is using, so for the first principal component, it would be let's call that 0.6 flipper length. Plus, so we'll call that 0.5. So 0.5 times body mass. We'll call that also 0.5. So that, let's call that negative 0.4. So negative. So this is how we calculate the first principal component. Does everyone see where I got that from? So these coefficients are basically coming from the size of these bars here. Yes, no? So the bar size comes from this comes from the PCA calculation, and we'll talk about this in a second. The way you can kind of think of these is these are kind of the contributions of a particular variable to a principal component. So in this one, build depth has a much larger contribution than the other three contributions. And in this case, build depth has no contribution to the third principal component, but it's really dominated by build length here. Does everyone get that basic idea, or do you want me to go over it again? And well, I haven't talked where these values come from yet. And I will say that this is actually one of the easily most hard subjects to kind of understand. So don't feel too bad if you're lost. Yeah, okay. So what I'm just going over is the contributions, like how are we calculating each of these values? So what we're doing, so this is basically a brand new variable. And you can imagine that we are just basically, this could be in a mutate statement. So what this is saying is that we're adding 0.6 times 0.6 times the flipper length. We're adding that to 0.5 times the body mass. and so on to produce our new Uber variable. So in this case, this is what's called a linear combination. So we are basically, so this is kind of how we're condensing multiple variables into a smaller number of variables. So is this becoming a little more clear? And to do this, to calculate the second principal component, you would do something similar with the value of these bars. Okay, so I'm going to go back to Allison's page because it discusses this better. How do we calculate, how do we know these values? So the principal components algorithm is trying to capture the variance in the data through these linear combinations. And so this is what's called kind of the variance plot. And the way to read this is that the first principal component is capturing about 70% of the variability in our data set. You kind of calculate one principal component and then you calculate the next one. So you're just really trying to account for the variance in the data. And so this first principal component captures about 70% of the variance. What percent of the total variance does the second principal component capture? Yeah, exactly, Brady, around 20%. So these linear combinations are kind of selected to capture the variance in the data. And you don't need to know exactly how this happens, but that's where these values come from. So you can see these are the coefficients for principal components 1. This is the coefficients for principal components 2, and so on. The other nice thing about this is that this analysis is guaranteed to produce new variables that are what are called orthogonal to each other. You can think of them as 90 degrees from each other. What does that mean? So that means that they basically correspond to different axes on this new plane of visualization. Yeah, so Sarah, this does not relate to the proportion that we designated earlier in the process. So this is something that's different. And it's not a dumb question. I'm not going to go over this code, but I like this plot better because it's the absolute value of the contribution. So you can see which one is kind of which variable is dominating over which principal component. Like which one is dominating over the second principal component. Yeah, exactly. So build depth, the bar here is much larger than the other ones. So this has a much larger contribution in terms of absolute value. Interestingly, so this one has build length as a top contributor, but it doesn't use build depth at all. And this has to do with kind of how these principal components are constructed. The main point really though is it allows you to compress a lot of variables. So you can theoretically compress like 1,000 numeric variables into 2 principal components. You need to generate that plot to understand what amount of variability the principal components are capturing. But usually, in the most cases with most data sets, about 90% of variability can be captured within the first principal components. I think it's something like 80 to 90%. But the main thing about it is it just lets you visualize a bunch of different variables at once on 2 axes. That was kind of a long explanation for this, but it is a useful kind of visualization. So I just want to introduce it to you. I will try to find a better discussion of this, and I will post it in the reading. But I just want to show you that you can basically calculate this as part of recipes. So who here is familiar with clustering or has heard of clustering? No one? Yeah. So clustering again is an unsupervised learning method. And you can ask the question whether there are natural groupings in the data. So this is, remember, this is different than, you know, the PCA step. So PCA lets us kind of visualize a lot of variables. The clustering helps us kind of discover whether there are natural groupings in the variables. So I'm going to go over Allison's example or Allison's GIF. Please tell me that it's okay. So with clustering, this is a method of clustering called k-means. And you specify the number of clusters that you think exist in the data. So these get basically kind of randomly placed on your in your data set. And basically, so you're calculating the sorry, this is going too fast. Let's wait for it to cycle back. But the idea is that you assign kind of a center of your clusters, and you assign like a grouping to the of each of the identity of each of these points belonging to a group is the ones that are closest to the center, to this cluster center. And usually you start at random. Sorry, Allison moves a little too fast. I should have tried to do this, but the main idea is that you'll get kind of natural groupings out of the data. So you've got your data, you've got these centroids. These are actually randomly assigned at first. And then through this kind of iterative process, we find the closest, the identity of each of these points is based on the closest center. But then we recalculate the centers every time. And it's this kind of backwards and forwards process. And in the end, hopefully, it helps us kind of discover whether there are groupings in our data. So, but the main idea is that, you know, you can start out with like kind of a random result, but like by just kind of recalculating these centers and the identity of these groups, you can basically discover natural patterns in your data set. And this is like, I literally spend a lot of time discussing this. I just want to show you that this is like an unsupervised method that we use very often. So like what we're doing, so we're going to supply penguins train. We're just going to select our species. So this is not in tidymodels anymore. So this is in base R. So we need to provide it with only numeric data. So we're going to take species out. And we're going to use this k-means function. So the main argument that we're supplying is we think that there are two groups in our penguins data. So what is in k-means model? And I will say that I am expecting that this is probably going to be one of the muddiest the muddiest talks. So I do apologize, but it is kind of I just really want to kind of introduce you to these unsupervised methods because they are very useful, and hopefully you'll like basically come back. So the final output is these are known as cluster centers. So we're saying the center of the cluster is at bill length 4.7 of the first cluster is at 47 bill length, 15 bill depth, 206, and then 5,129. The centers of the second cluster are these locations. And each row of our data gets assigned to our two different groups. So it seems like we've basically used this, and it's divided our data up into what's roughly, so our first cluster contains 84 penguins. Our second cluster contains 109 penguins. So one question that you might ask is, so this is a kind of a natural grouping. How does this compare with our actual species labels? So we need to, this is the third function in Broom that we didn't talk about. It's called augment. And what augment does is it will, in our case of the k-means model, it will add these clustering assignments, so this grouping assignments, to our original penguins trained data set. So if we look at clusters, we'll see that it's our original data set, but we have this additional column called dot cluster here. And these are the grouping assignments that were automatically assigned by the clustering algorithm. So if we actually do a crosstab, well, this is just to kind of say, so these are the groupings found by the automated algorithm. These are the true values of that. So if we do a crosstab, so we can just basically do a crosstab of these two using our janitor table function. And the way to read this is that our algorithm predicted 1, and the true identity of this is Adelie. So how many did our algorithm predict 2, and where the species is actually Adelie? Yeah, exactly, 99. So this is much like any 2x2 table that you've seen, except that these labels are not, and this is an important thing to note, is that these are the natural groupings found by the algorithm. This is not a prediction. But you can see that it seems to be that we can find kind of a good, a natural grouping that basically assigns the majority of our Adelie penguins into group 2 and the majority of our Gen 2 penguins in group 1. So that's just kind of, and again, I didn't have that much time to kind of put this notebook together. So again, I apologize if it's confusing, But the main point is that we can kind of visualize our data. We can squish a lot of different variables into kind of 2 dimensions to visualize it. We can basically apply an automated method, and we just give it the number of groups, and it's going to try to discover the number of groups in our data set. Actually, so what I'm going to show you now, I think maybe this will make things a little more clear. So I'm going to jump down to trying different values of k. So this is where you can see that this is where it becomes very, the map, per and map become very powerful. So what this is doing is it is running, we're going to, our input is going to be basically a single column data frame. And we're going to range over the values of k from 1 to 5. And so our first step is that we're going to map over these values. And we're going to say, okay, so so for k is equal to 1, this is going to be k-means. We're going to run the k-means algorithm, and then we're going to supply the value of 1. Then we're going to basically apply tidy, glance, and augment to kind of produce our table with list columns. So this is actual k-means analysis, but these are the different versions of the data set, or the different versions of our metrics. And remember, glance gives us kind of metrics on our data set, and then of our method. Tidied, in this case, I can't remember, but we're mostly after augmented. So what we're going to do, so the main thing I'm going to work with is this, is the augmented data. So let's look at that. So I'm just going to use unnest to pull out the augmented columns. So this is confusing when you first kind of look at it, but you can see that we've got k. We basically ran our clustering algorithm for 5 different values of k. And each of these is going to have the number of clusters that we assigned, and then each of these rows is going to be a cluster assignment. So let's take a look at that. So I'm going to go down to here. So what's going on is, like, the algorithm is trying to find these natural groupings in the data among all of the different variables. We're only visualizing body mass and build depth here, but you can see what the identities of the clusters are. So here we told the algorithm we only wanted 1 cluster. So it only returns 1 cluster. Here it's returning 2 clusters, but you can see, it's all, and this is kind of a hint that the separation is not always kind of found within, like, the 2 variables that we're visualizing. This is 3, so it is trying to divide it up into 3 different clusters, even though 2 seems to be kind of the natural grouping. We know that there are 2. If you put in 5, this is what the clusters look like. So it is a very, in a way, it's a very kind of dumb algorithm, because, like, you are kind of you're saying, try to find 5 different groupings of our data set. And you can see it's not doing a great job if our number is high. Does that make a little more sense as to what's going on? kind of, yeah. And again, the main purpose of this is really kind of to help us kind of discover whether there are natural groupings in our data set. Yeah, so like it's really trying to find these kinds of divisions in our data set. So if we visualized, let's visualize like bill length. You can see that these combinations, like here, like for number of 5 clusters, like it's not really finding this kind of separation in the data. So like you can see all of these clusters are overlapping here. And again, it's trying to find this kind of division among all of the different variables that we're putting into it. Here too bill depth versus bill length does seem to be a pretty good kind of separation between our two groups. I just really want to kind of show you this because this is a very useful technique. Actually, a lot of market analysis is basically done on clustering. Have you ever heard about market segments? Anyone? Yeah, so market segments are kind of, this is the interesting thing is that, so like say you take like, you know, everybody's shopping cart and shopping habits. Okay, see you later, Eric. You know, there might be a group of people who really, who buy diapers. So what the algorithm is going to do is it's going to kind of look through the data and it's going to say, oh, look, we found this group that seems to be focused on buying diapers. So that group is called a market segment in this case. So basically, if you've heard these kind of personas that are applied in marketing, this is kind of where they come from, is basically they're trying to find these natural groupings in the data and discover what the identity of these people are. So it's kind of an automated approach, but then you have to kind of go back and you kind of investigate these groups that the algorithm has kind of discovered. So for example, this group might be moms, and this might be gamers, for example. So like, you know, these people, I'm really trying not to be sexist here. So, I mean, let's say parents. So this group buys diapers. This group tends to buy video games or something like that. But it is a technique that's actually used quite a lot. And, you know, I spent my master's thesis actually kind of understanding how these things work. But I don't have enough time to kind of go through them. So let's wrap up with the logistic regression. And this is just to set us up for not only the assignment, but to show you kind of the basics of logistic regression. So what we're doing here is we're just taking our penguins train. I guess I could have used a recipe to do this. But I am just using a case one statement. So when species is equal to Adderley, I want it to be true. And when species is equal to Gen2, I want it to be false. And then I'm going to take out the original species variable. So this is our new variable. It is a Boolean variable, also a 0. And because Boolean underneath it is a 0, 1. So it is a dichotomous variable. So we can use this dichotomous variable in our model. And to run a logistic regression model, we use the LM function again. We put in our formula. So our outcome here is Adderley. And then we're using the rest of the numeric variables. Our data set is penguins train 2. And then to do the logistic regression, we need to provide this argument of family binomial. And I don't know, like when I was running it this afternoon, it was giving me, I think they have changed the behavior of this, because it used to be to specify a logistic regression, you needed to provide this argument. I think it is now smart enough to realize that if you give it a dichotomous variable as your outcome, it will basically build a logistic regression. So I know most of you haven't seen logistic regressions, but the main one of the main outputs of a logistic regression model is the probability, is a predicted probability. And we'll get into that in a second. So we've ran our model. Now we're going to use the augment, and we're going to put we're going to use data as equal. We're going to augment our Penguins train 2 data. We're going to add this type predict column. So this is basically we're specifying that we want the predicted probability. We don't want the logit value. But the main point is that we're going to get a probability out of it. So, and then I'm just adding the species column back into this to make it easier. So let's look at penguins augmented. So you can see there is this column. Wait, I need to figure out what's going on with this. This is not the predicted probability. I will go back and fix this. I think there is something wrong in my LM fit, so I'm going to fix this, but I do want to show you if we visualize this. Yeah, this is wrong. I need to figure out what's going on here. But again, so our predicted probability, this should really range from 0 to 1. I need to, again, I'll go back and figure out what's going on here. I think it might be because I used a Boolean variable. Let me try this. So let's take a look at penguins augmented again. No, what is going on here? Okay, I will figure out what's going on with this. Ideally, what this should be is the probability that a species is audily, or that a penguin is audily, based on our logistic regression model. And so if our probability is high, then we predict that penguin is going to be is going to be an audily penguin. If that probability is low, then the probability is going that the species is probably going to be Gen 2. So I will go back, and again, you don't have to let me go back and fix this. I'll address it next time. I apologize for that. But next time we can kind of talk about the probability distribution plot that is generated as part of logistic regression. This is the last little bit. So this is optional, but this is, I had the question of how do you run multiple models on a data set? So what I'm making here is a data frame, and I'm using a vector of characters. So I'm specifying the formula here as a character. So this is a vector of characters. So if we just run penguin formulas, you'll see we can do this. So my function that I'm going to apply is going to take this value, this character value, and you can use the as.formula to change that into a formula that you can put directly into LM. And then this is just me running the model. And so you can see that we've ran all three of these different models. with different covariates, covariates on our model, and that we can basically get things like R² values out of it. So you can see that bill length plus bill depth has the highest R² here. And then if you unnest the tidy output, you can get the coefficients. So hopefully that answered that answered that question about how do you run multiple models. Again, so don't worry, there's not going, just basically for your assignment, just basically start exploring the Nhanes data set with your outcome in mind. Try to kind of meet before class, and office hours might be a good time to do this. Just meet before class and kind of decide on a list of covariates and how you're going to divvy them up among the model. And I think there was a question. So your models can have multiple covariates. There's a lot of them. So, you could run 2 models, like one with 2 covariates and one with 3 covariates. I'm going to leave the complexity up to you because I think it's more, I don't want to stress people out. So that's all I have for today. So I will say just kind of bathe in your confusion, and I will try to find some literature kind of that talks both about clustering and principal components analysis.